All right, so today we're going to finish up our genetics notes by looking at biotechnology. So, this is kind of a funny cartoon about bioengineering and biotechnology. So basically what we're trying to do is take DNA and combine it in new and different ways. So biotechnology is the use of microorganisms, cells, or cell components to make a product. So what are some of these products that we can make? Well, we can make things like foods, vaccines, antibiotics, vitamins, and even things that undergo biodegradation. Um, Biotechnology does include things like selective breeding. So, when we pure breed dogs or even cats or other animals, that is a form of biotechnology. So, how do we do this? Well, a typical molecular biology technique is to get into the nucleus, take a look at the DNA, find the different genes, and then manipulate those genes for our benefit. So what we're going to be doing is kind of taking a look at some of this and kind of going through some of the different techniques that are used. So when we actually talk about genetic engineering, we are talking about manipulating an organism's genome. Whether it's going in with, like the little needle that we just saw there or here, and manipulating it. But we can also alter plants and animals and even microbes to our benefit. We did this actually with corn. Corn's original ancestor looks nothing like the corn plant that we have today. It was actually more of a grass. But we selectively bred it so that we had plants with kernels that had high nutrition value to it. Again, we look at different animals. We want to breed animals that we use for food sources that, gain weight easily, that are healthy, that produce a lot of meat. And so we do that, in the world as it is today. We can also use this to help to correct genetic defects that we have in humans. So genetic technologies were also used to identify individuals, and this was part of a project to help identify certain victims of the Holocaust. We could actually use that to find the family members that maybe had been missing and were part of, a lot of the Holocaust people were never identified. So they're actually using this now to try to find their surviving family members and actually give them a name and the honor that they deserve. There's also different projects that help you trace your ancestry. I mean, you've seen the Ancestry.com ads on TV, and they have it where, I thought I was this and now I'm that. But it also can be used for the diagnosis and epidemiology, so the tracing of how diseases form, because diseases do change over time as well. When we look at plant biotechnologies, we've got a couple of things that we can do. First of all, we can have them undergo mitosis, so in other words, we can clone the different plants. Or we can use meiosis, so breeding plants for their specific traits, like drought resistance. So this can be plant to plant, where we're teasing out some of the similarities that we like and that we want to keep in them. Or it can be transgenic, so we introduce new genes into the plants. I actually did some work with this in my undergraduate studies in college. We actually were combining spinach cells with tomato plant cells and creating new plants and trying to see which genes were favorable in that new plant. And it was really interesting work. Difficult because it was early, but very interesting. All right, so a couple of definitions here. When we refer to something as being transgenic, we're talking about an organism that contains genetic material into which DNA from an unrelated organism has been artificially introduced. So that can be, combining, fusing cells and nuclei. It can be snipping out genes and inserting them. There's a number of different ways that we can do that. A marker gene. This is what we use to determine if that nucleic acid sequence has been successfully inserted. We can cause that little marker gene to create maybe like a fluorescence, something that we can see and identify and say, oh yes, that has actually been uptaken in that new cell. So overall, our goal for plants has been to improve our agriculture, to improve the plant's resilience, the resistance to herbicides, the harvestable quantity, so the crop yield, so to speak, the flavor, the nutrient value or the nourishing quality, and the spoilage resistance, so how long we can keep it before it goes bad. When we look at animal biotechnology, we've got mitosis, again, cloning of animals. And in meiosis, we can actually breed animals for specific traits like milk production or physical characteristics. Again, I go back to, dogs because it's something that we do. There's certain characteristics of different dog breeds that we want to have if we want that type of dog. So again, just like plants, it can be animal to animal, so same species, or it can be even transgenic, introducing new genes. So when we look at something called a bioreactor, these are living organisms, and we typically use bacteria, not multicellular organisms, but we can use bacteria to synthesize useful substances or break down harmful ones. So, we can actually take genes from animals, insert them into bacteria, and have them produce what we want them to produce. Or break down the harmful substances that we don't want. So with animal biotechnology, our goal is again to improve agriculture and our food supply. The size of the animals for more meat, the amount of product that we are making, let's say for milk. Also human medical products. We can actually grow extra organs or organs that are going to be more suitable for humans with organ transplants. Biochemical products or even proteins, things like insulin. We can actually do this. If we don't have human insulin, we can actually use sheep insulin, and we can actually insert the gene that creates sheep insulin into bacteria and have them create lots and lots and lots of this insulin for use by diabetics. All right, so for medicine, from a medical perspective, what we are trying to do with all of this bioengineering is to cure or eliminate human diseases, create immunity to different viruses, think like HIV, AIDS, swine flu, bird flu, all of that kind of stuff. And if in lacking that, create new medicines that will help us fight some of these things that we can't create vaccines to. So how do we do this? How do we actually work with this? Well, we have some common techniques. There's 4 different techniques: southern blotting, gel electrophoresis, PCR, which we'll talk about because that's an abbreviation, and recombinant DNA. So let's look at southern blotting first. Basically, southern blot is a method used in molecular biology for the detection of a specific DNA sequence in DNA samples. So what it does is it combines the transfer of electrophoresis separated DNA fragments to a filter and then detection of specific fragments by hybridization. So this is what is happening. So we have the unlabeled DNA cut. So we basically take DNA and we've cut it up and we run it on a gel electrophoresis. So then what we do is we put the stack of paper towels with the nitrocellulose paper on top of the gel, and then we remove that. So that literally we're like blotting it. If you think of when you blot stuff, you push something down and you pick it up. Well, in this case, we are picking up the DNA. And then we take that labeled DNA probe and then we hybridize it. Gel electrophoresis is where we actually separate and analyze the macromolecules. Typically it's DNA, but it can also include RNA and proteins. We look at their fragments based on their size and their charge. This is what creates what we call that DNA fingerprint that we use in like forensics. So what we do is we put the DNA samples here, and what we've done is we've actually cut up the DNA with an enzyme, so not with scissors, but we use a chemical. And the DNA actually moves across the gel, and the longer bands don't move as far. Think of big things. They move slowly, they don't move as far, and it works the same way here. So the bigger pieces of DNA stay closer to the original, whereas the smaller pieces of DNA will move farther. They can move more quickly through the gel. PCR. PCR is basically, I kind of call it the Xerox for DNA. Okay, it's the copy machine. Because what you do is you amplify a single copy or a few copies or even a piece of DNA across several orders of magnitude. So you have this teeny bit of DNA, and you make enough of it so that you can actually work with it. So we are talking about thousands, even million copies of that particular sequence. So what we do is we take that little piece and we just keep photocopying it and photocopying it and photocopying it and photocopying it, and we end up with many more copies of that DNA. Recombinant DNA, which is sometimes abbreviated rDNA, is what we do is we actually combine genetic information in one organism, so like molecular cloning. So what we do, this would be a great way to, like, I was talking about inserting that insulin gene. This is actually how they do it. They create this recombinant DNA. So they're bringing together genetic material from multiple sources and create sequences that would not otherwise be found. So what we do is we use something called a plasmid, and bacteria have these plasmids. These are extra copies of their DNA. We can actually cut across or cut open that plasmid and insert a gene. Okay, so that gene gets inserted into these cells, and the ones that have the gene inserted then are selected out because that's the ones we want, and they will actually produce that protein for us in large quantities when we grow those bacteria. So what are some of the challenges with this? Because these are technologies that exist, but they're not perfect. Well, first of all, getting the gene into the correct cells. Sometimes getting it to the right place is not as easy as it sounds. Getting the gene so that it expresses correctly. You have to worry about orientation of the gene. You also have to worry about regulation of the gene. Controlling the virus vector. There are ways that viruses get introduced that we can't always control. Think of Zika right now. How do we control those mosquitoes? One of the things that they're actually looking to do is to genetically engineer a mosquito that when it gets infected with Zika, it is no longer able to reproduce. So that way then it's not going to bite, it won't pass it on. There are also ethical issues with this, and some of these are coming to the forefront. For instance, how are they going to affect our environment? Should we be messing with the genetic code? How can we, should we be spreading of genes to these other organisms? So let's say we have people who don't want to have food that's been genetically modified like this. How do we ensure that we don't have cross-contamination of plants? Pollen travels a long way. And the last one is kind of like, who's going to decide this? How are we going to figure this out? So, last little bit here, we have a cartoon. We've genetically engineered a tuna exactly the same diameter as our cans, which is kind of funny. So all you have to do is chop it up like you're slicing a pumpkin roll or another roll and slide it right into the cans. Ha ha, wish it was that easy. All right, so that is it for biotechnology.